Today, we're going to tackle a really fascinating scientific exercise on Parkinson's disease. This is all based on some great material from Hanadi Nisrallah, especially for the students of the SE Lebanese curriculum. We're going to solve this thing together, one step at a time. Okay, let's dive right in. The game plan is pretty simple. We'll use the documents provided to really understand the disease, check out a potential treatment, and then, like any good scientist, draw a conclusion based purely on the evidence. So, first things first. Before you can even think about a solution, you've got to wrap your head around the problem. So, what exactly is Parkinson's disease?
Right off the bat, the source material gives us a solid scientific definition. Now, there are two key words here you got to pay attention to. Progressive, which means it gets worse over time, and neurodegenerative, which tells you it's all about the breakdown of nerve cells in your brain. And at the absolute heart of this disease is a chemical called dopamine. You can think of dopamine as this crucial messenger that lets your brain talk to your muscles, keeping everything moving smoothly. What the source tells us is that with Parkinson's, the brain just starts making less and less of it. And when those dopamine levels start to drop, well, it has a very real physical effect. The signals from the brain to the body get all jumbled up, and that leads to a loss of motor control, which is really the central physical challenge of this disease. So, what does this loss of control actually look like? The source points to those classic symptoms you might have heard of: tremors, really stiff muscles, and just a general slowness in movement. But it's not just physical. It can also affect your mood and your thinking, leading to things like depression. All right, so I think we've got a pretty good handle on the disease now. The next question is obvious, right? What can we do about it? And that's what brings us to the next part of this exercise. So now we're shifting gears. We're moving away from just defining the problem and starting to investigate a potential solution. The source material introduces us to an experiment that's all about a drug called selegiline. Okay, so the source tells us this molecule is used as a treatment. But just knowing the name isn't enough, right? As scientists, we want to know how. How does this thing actually work to fight the disease? And here's the setup for the experiment. It focuses on something called the synaptic cleft. Just picture it as this tiny little gap between two nerve cells where all the messages get passed. The experiment itself is super straightforward. Measure how much dopamine is in that gap without the drug, and then measure it again with the drug. Okay, this is where it gets really good. We're at the heart of the whole exercise now. It's time to actually look at the evidence and analyze the data from that experiment. And here it is, the raw data. This table shows us two different situations over a very short period of time. That top row, that's our baseline, what happens to dopamine on its own, with no drug. The bottom row shows what happens when selegiline is in the picture. The important thing to notice is they both start at the exact same level, 100 micromoles. And this is where you can see the story the numbers are telling us. I mean, it's night and day. On the left, without the drug, the dopamine just vanishes. But on the right, with the drug, that decrease is so much slower. You can see it's having a huge protective effect. I mean, just look at that top row again. The drop is absolutely wild. It goes from 100 all the way down to 0 in just 5 milliseconds. It's completely gone, wiped out. But then you add selegiline, and it's a completely different story. The drug is like a shield, seriously slowing down that breakdown. After the same amount of time, more than half of the dopamine is still there, ready to work. So really, the final numbers tell you everything you need to know. Without the drug, you're left with 0. The communication channel is basically shut down. And with the drug, you've still got 55 micromoles available to send signals. That difference between 0 and 55, well, that's everything. It's a huge deal. So we've looked at all the evidence. Now it's time for the final and most important step of our exercise. We need to put it all together and draw a conclusion. What can we actually say selegiline does? Based on our data, the conclusion is just undeniable. Selegiline works by protecting the dopamine that's already in that little gap between the neurons, slowing down how fast it gets broken down. It's basically keeping the message alive for longer. And you've got to remember, this isn't just about some numbers on a chart. For a patient, this is potentially life-changing. When there's more dopamine available, it means better communication between neurons. And that can lead to better motor control, fewer symptoms like tremors and stiffness, and at the end of the day, a better quality of life. And this ties it all together so perfectly. What we concluded about selegiline lines up exactly with the goal of treatment mentioned way back in the source material. The drug helps manage the disease by You guessed it, attenuating the symptoms.
And that pretty much completes our walkthrough of the exercise. We defined the problem, we analyzed the data, and we came to a really solid conclusion. But it does leave us with one last big thought, doesn't it? A drug that manages symptoms is absolutely critical. But what's that next step? What's the next scientific frontier that could take us beyond just managing and maybe towards a cure? Definitely something to think about.